Good evening, everyone. Dr. Minal Bhujangra Wagmar is here. In the series of diseases of field and horticulture crops and their management, we are now seeing the pulses crop. In that, our first crop is gram. So, coming to the so our first disease of chickpea is a gram wilt. Causal organism is a Fusarium oxysporum, former species Ciceri. This pathogen is basically soil-borne fungus. In wilt, what happened? That pathogen will attack on the vascular tissue of the plant. Vascular tissue is consist of the xylem and phloem. Xylem helps for the conduction of the water and phloem helps for conduction of the food. Once vascular tissue is damaged, the transportation of the food and water supply to the plant get stopped, and due to that, uh, wilting appearance and drying appearance comes to the plant. In this, leaves turns yellow and ultimately get dry and fall off. And in case of the defense, like pathogen, when attack on the vascular tissue, plant also try to defend itself and it create the dilysis formation, which try to restrict the movement of pathogen from vascular tissue to throughout the body of the plant. And ultimately, due to vascular tissue damage, stems may turn to the brown and black, especially near to the base, and plant will die with, due to the wilting, that is, drying appearance and not proper supply of water will take place. So ultimately, plant will die. Coming to their life cycle, in this disease, basically three major spores will form, that is chlamydospores and macroconidia and microconidia. In this chlamydospore is a resting spore or overwinter spore. Whenever unfavorable condition will come, it will survive in a soil debris, and when favorable condition will come, it will again germinate it and host is available, then it will cause the disease and lead to the infection cycle. And other spores are formed, that is macroconidia and microconidia. Coming to their management, as we seen, it is a soil-borne fungus, so deep summer plowing is needed. In this, the soil which present at the lower layer, it will come at the upper surface, and if pathogen debris or spore structure is present inside such soil, it get destroyed by the sunlight. We have to go for crop rotation to break down the host availability, continuous host availability to the pathogen to break down its life cycle. Use a disease-free seed means like resistant variety we will use for cultivation purpose. In this, to enhance the soil quality, we use the FIM 10 to 15 cart load per hectare. Other some seed treatments we see that carbendazim and thyram 2 gram per kg of seed and trichoderma viridi 4 gram per kg, and so Pseudomonas fluorescens, that is 10 gram per kg, we can use for a seed treatment. Again, here are same again thyram plus carbendazim together 1 gram plus 2 gram per kilogram of seed. We can provide such a seed treatment. Mainly, other than this, proper drainage and proper irrigation channel maintenance should do to control the wilt disease of gram. Another important disease of the chickpea is grey mold of gram. Causal organism is a Botrytis cinerea. In this, we can see that water-soaked spots on the form on the pods as well as seed and on leaves too. This turns brown and blackish in color, and leaves become a yellow and drop and flower become a rotteny moss. In somewhere, wilting and patches of dead plant and seedling of damping of kind of symptoms also appear and eventually lead to the plant death. Here, water spots has been formed on the seeds. Here we can see that, and resulting that seeds get shriveled and number of seeds also reduce and size also get reduced and quality of the seed also get reduced. Coming to the life cycle, hard structure of sclerotia blackish in color present in the stem pith and infected plant part, and it survives in a soil and crop debris too. And some another spores, that is conidia, get released from this crop resident sclerotia and lead to the another infection cycle. And grey fungal growth and floration on the stem takes place, and this spores formed on the infected plant can initiate the secondary spread of this disease. This conidia will lead to the secondary spread of the disease. Coming to its management, avoid the excessive vegetative growth. It means controlled dose of nitrogen should be applied to avoid the excess vegetative growth, which generally attracts the pathogen of Ascochyta blight. Intercrop with the linseed that pathogen can divert as well as enhance the other income of the plant farmer. Avoid the excess irrigation. We have to maintain the irrigation channel, and deep summer plowing again helpful in such a in this management because it will help to destroy the pathogen spores which present inside the soil with the help of sunlight. Again, some coming to the seed treatment of fungicide, carbadoenzyme and thiram 1:1 in 1:1 form, 3 gram per kg of seed is recommended. Captain to 5 to 6 kg per hectare at 15 days of interval also recommended. Coming to the spray, mancozeb of 3 gram per liter, that is 0.3%, also recommended as a foliar spray. In this, first spray will done when initial symptoms are appear, and second spray will done after the 15 days of first spray, or if symptoms are there, then only we will do, if needed, then only we will do second spray. Coming to the next disease, Ascochyta blight, that is Ascochyta rabi. In this, all plant parts are affected, but mainly water soaked lesions will form, and this symptom shows the small circular brown spots on the leaves. We can see small circular brown spots are coming on the leaves, and this ultimately lead to the dry and necrotic tissue inside that leaves, which reduce the photosynthesis and ultimately lead to the losses of the yield. In this, size of seed get reduces, quality of seed also get reduces, and pods also get reduced in size, and number of pods also reduces. And in severe cases, entire plant get dry up suddenly, kind of wilting appearance will come in a severe infection case. Coming to its life cycle, there are two life cycles are occur in such disease, that is sexual life cycle and asexual life cycle. Sexual life cycle leaded by the ascospores, that female hypa and male hypa get fused and lead to the formation of pseudothecium, and this pseudothecium leads the ascospores. When favorable condition is there, sexual life cycle get continue. When unfavorable condition is there, that pycnidium will release the conidia, and that conidia gets spread by wind and rain, and infection, secondary infection lead by the conidia. So coming to the management, so the disease free seed, go for the crop rotation, intercrop the seed with the wheat, barley, mustard and linseed to avoid the infestation of the infection of the pathogen completely on the main crop and enhance the other income of the farmer. Coming to the fungicide seed treatment, carbendazim 1 gram per kg of seed is applied, and hot water seed treatment also help to manage at 52 degrees Celsius for 10 minutes given by Jensen earlier. Again, a spray of the mancozeb at 0.25%, that is 2.5 gram per liter if the infection is noticed during the growth period, and other spray of vegetable sulfur at 0.23, that is 2.3 gram per liter of water is provided. So if you like this video, please like my channel and subscribe my channel and share the video. Thank you so much.